Hello everyone. So this is me, Ishan, talking about science and tech once again with you all. And today, what we're going to do is we're going to talk about some basic concepts. So this is going to be a series of videos, and I want to start from first from the basics because my belief is that the concepts are of utmost importance in science and tech, and rest of the things you will not be able to understand if you are not thorough with the basics. So again, there is a lot of concepts to be covered, but I'll try and cover the most important ones, and then the rest of the concepts are give a glimpse of it, and then you can read more about it. And in the next series of videos, in the next set of videos, when we talk about certain things in depth, then maybe I'll revisit those deeper concepts. But right now, we're just trying to cover the basics, the essentials basically for every topic in science and tech. So for space, we'll be doing launch vehicles and engines, and the element formations in the universe, things like that. Then for biotech, we'll be doing DNA, RNA, nucleus, uh, how the chromosomes are there. So, for health, we'll be doing some kind of immune system, vaccines. So, similarly, we'll be proceeding for nanotechnology, nuclear technology, the basics, right, for computers, IT, communications, and robotics and AI, all the basic, the basic overview with basic definitions. Um, so that's how I'm going to proceed, and I'm going to start with space technology. Uh, so let's get started. Let's be quick, and let's try and cover this as fast as we can. All right, so first let me look at space technology, and let's start right away with launch vehicles. Now, uh, you must have read about different satellites that are launched in space. So basically, what are launch vehicles? They are as simple as the words itself, vehicles which are used to launch objects in space, right? So that's the simple thing about it, and they are also called as rockets. So sometimes, people get confused. What is a rocket and a launch vehicle? They're just essentially the same thing. There's a difference between a rocket and a jet, which we will come to that. We'll come to that when I explain jet engines. Right. So when you look at rocket or a launch vehicle, what usually happens is that there are two parts to it. Okay. So one part is the one which is carrying the payload. So if you look at the PSLV over here, there'll be a part which is carrying the payload. Okay. So payload is usually your satellites. So satellites they could be your Mars orbital missions payloads, or they could be Chandrayaan, anything. PSLV did not carry, it carried the first Chandrayaan. So anyway, it could be any satellite basically, and the rest of the body is just fuel chambers. Fuel chambers. Now this is very important to understand because if you look at the structure of the rocket, most of most of it is fuel chambers, right? And what is there in the fuel chamber? First of all, there are different stages. Okay, so there is so in PSLV, let's suppose in PSLV, we're talking about there's a solid stage, then there's a liquid stage, then again there is a solid stage, and then again we have a liquid stage, right? There are different stages. Now, what are these stages? We'll try and understand. Before that, let me tell you that in different launch vehicles, uh, there'll be different types of stages, and there's a different reason for every launch vehicle to have those kind of stages. Okay, so in each of these stages, first of all, in each of these stages, what happens is there is a fuel, and then there's an oxidizer. So what is the fuel? Fuel is something that will burn, right? And what is an oxidizer? It is thing which will cause the burn, which will cause the burn, right? So why do you need an oxidizer? Because you don't have oxygen in outer space, and you cannot do combustion of fuels without oxygen. So that is why you need oxidizers, along with fuels. So every fuel chamber has this combination. One is a fuel, and then there's an oxidizer. So every fuel chamber will have these. Now, uh, I just spoke about solid liquid, right? So why are these solid and liquid? Because solid stages have solid fuels, liquid stages have liquid fuels. Okay, liquid stages have liquid fuels. Okay, and then there's one more stage, cryogenic. We'll come to that. Okay, but before that, let me just give a brief about the kind of differences that we see in solid and liquid fuels. So what happens is in solid fuels, if you have a solid stage rocket and if you have a solid fuel, mostly these are used in the first stages, the initial stages to power the rocket, because they are usually uncontrolled. And once you ignite a solid fuel, there is no way to stop that reaction. So it is an uncontrollable reaction, and it is highly powerful. So mostly you need a lot of thrust in the initial stages. So you want to use solid fuels for that, right? And then in the next stage, what you do is you use liquid fuels. So in liquid fuels, um, they can be slightly better controlled than the solid fuels, because if you have a liquid, then you can control the inlet and outlet for that, and that is why they have better controllability. But at the same time, it's difficult to carry them, because if you have a liquid in a tank, and if you move that tank at higher speeds, then what will happen? The liquid will try and juggle a little bit. So the rockets are moving at a very high space, very high speed. So what happens is liquid fuels become a little bit of there's a little bit of difficulty in carrying them, but again, there's a controlled reaction, so that is a good thing, right? And uh, then what happens in cryogenic? Now you must be curious as to why I've not written solid, liquid, and gas. Now if you have a gas, let's suppose that we did not go for we did not know what cryogenic is. We did have a uh, gaseous fuel chamber, so that will mean that it is having gas fuels. Now imagine if you have to store gas, you'll need such big tanks, because gas will require a lot of volume. That's the definition. It needs a lot of volume. So, do you really have that much of space? No. So what you try and do is you try and compress this gas. You try and compress this gas at very low temperatures. So cryo means low temperatures. No, so cryogenic means they're generated from low temperatures, right? So when you try and compress some gases, we'll see what those gases are. Try and compress it at very low temperatures, they become liquid, right? And when they become liquid, it's easier to store them, um, as compared to if they were gas, and they are lighter also. So these gases are always lighter, right? So if you convert them to liquid, again they are lighter. And remember, if I told you in the beginning, most of the structure of the rocket is just fuel chambers. So sometimes what happens is uh, rocket has 90% of the weight in the form of fuel chambers. So, just 10% is what your actual payload is. And most of the times, the rocket is pulling its own fuels. So that is what is happening, because most of the parts are just fuel chambers. So what do you want to do? What do you want to do? You want to reduce the weight of these fuel chambers, right? This is where the cryogenic engines, the cryogenic fuel chambers, they become very, very important, because then when you have a cryogenic engine attached, you're essentially trying to reduce the weight of the rocket, right? And that's very important for us, because most of the weight of the rocket is just fuel chambers. All right. So I gave you the example of PSLV, where there are four stages: solid, liquid, solid, liquid. So there's no cryogenic stage as such. PSLV is known as the workhorse of ISRO because of its grand success. Now it's a very, very old launch vehicle being used for more than 2 decades, and it has had a lot of success. Right. So uh, launch vehicles, they are used to launch objects in space. They have two parts. One carries the payloads, other carries the fuels, right? Fuels will always have an oxidizer along with them. And materials, you can refer to it. You need strong materials, right? Because you'll have these launch vehicles pass through different layers of space, and there are different conditions in each layer. So you want a material that can withstand all those different conditions, right? So usually alloys of iron, nickel, cobalt, titanium, or Kevlar is the most, mostly used alloy. So all these things, you can remember uh, for the materials that I used, right? So let me just come back to the solid, liquid, and cryogenic thing that I was talking about. So fuels, they can be solid, liquid, or gaseous, but gaseous fuels are mostly used as cryogenic fuels, and they're the best, followed by liquid and solid fuels. So I gave you the comparisons, and you know why cryogenic fuels are considered the best. Right. So these combinations, if you look at these combinations, so these are examples of combinations of fuel plus oxidizers. There's a fuel here, then there's an oxidizer here. Similarly, here you have a fuel plus oxidizer. Similarly, so for solid, you have these. There's chemical full form to this, which you don't need to know. You don't need to remember also. If you just remember STPAPB in exam, you can figure it out. If they have written the full form, you can figure it out. It's STPP or UDMH for liquid, right? So beyond that, you don't need to, mug up all these names, chemical names. More important is the concept, right? That there's a fuel and then there's an oxidizer. Now, these liquid engines, they were developed um, based on the French Vikings engine, and they were named as Vikas engine in India. Vikas engines. They are the liquid engines. All right. So if you look at whenever you read news and if you look at the different stages that they're talking about, mostly what they'll do is they'll write S11 or let me say S100 or L110. No, so usually what they're trying to tell you is it's a solid stage which is carrying 100 tons of fuel, or a liquid stage which is carrying 110 tons of fuel. That is how you read it. Just so that when you read it next time in the newspapers, you will have a little bit of understanding for this. All right, so now let me come back to cryogenic, which why I was interested in, and ISRO is interested in because of its advantages. So what we are trying to do is we're trying to use liquid hydrogen as a fuel and liquid oxygen as an oxidizer. All right, that is happening in the cryogenic stage, and we are currently using CE20 engines, right? So 20 tons of fuel, and this is used in GSLV Mark 3. So now it's called as LVM3, right? That is where we are using cryogenic engines. But what happens is ISRO says that okay, we have a cryogenic engine. Now what about we make it even better, right? So they want to make, and they are in the process of using this new thing which is called as a semi-cryogenic engine. Now, what happens in a cryogenic engine is that instead of liquid hydrogen, you're using kerosene, right? Plus again liquid oxygen. Okay, so the fuel changes and the oxidizer remains the same. Now, because you're using kerosene, kerosene is even lighter, and it has a better storage performance compared to hydrogen, right? So this again becomes better than the cryogenic engine. Now, what uh, what ISRO is trying to do is trying to replace the liquid stages with the semi-cryogenic ones. Because if you reduce the weight of the rocket, once again you can add heavier payloads, because that is the problem. Because usually, why are your launch vehicles not able to carry big payloads? Is because most of your weight is just fuels. So if you remove that weight, you can compensate for it in the payload part of the rocket. So you can put more stuff over here in the uh, upper body of the rocket, which is carrying payloads, and this part will become lighter, which will make it easier for ISRO to send heavier payloads. So they're trying to replace liquid with semi-cryogenic. It's all under process. A lot of testing has been already done. It has already been uh, successful. But slowly and steadily, we'll see that this will come up in the design of rockets also. Okay, so let me just I've shown you PSLVs already, and let me show you LVM3. If you want to have a look at LVM2, no, not LVM2, it's called as GSLV Mark II. So if you want to have a look at that, you can go and have a look at that. It was not a very great success, but maybe they'll use it some some time in the upcoming missions. So you can just have a look at that. Mostly what we're using right now is LVM-3 or GSLV Mark 3. This is the old name, GSLV Mark 3. Now it is called as Launch Vehicle Mark 3. So it has three stages. Again, it has three stages. So these two that you see here, these are solid stages. Then there's a liquid stage over here. Inside this, there's a liquid stage, and then there's a cryogenic stage over here. Okay. So the recent success of Chandrayaan 3 is credited to LVM-3 rockets, and is the most powerful that we have right now. And the special features of this rocket is the cryogenic engine that we are using. All right. So yes, LVM-3 is currently the most powerful, but ISRO is under uh, process of developing newer launch vehicles. So let me just give you a glimpse of those. So there's something called unified launch vehicles, which is you know a modular, expandable version of all the launch vehicles. So what they're trying to do is they're trying to remove all the PSLVs, GSLVs, and put just a unified launch vehicle, which will be a modular one. So you can just you know um, modify it as and when you want changes, right? So it will be very easy to modify uh, for your specific needs, right? So that will be your unified launch vehicle. And there's another thing that ISRO is trying to develop is reusable launch vehicle, looking at SpaceX and other US startups and other developments in the global space industry, as well as the need for sustainability, right? Because a lot of times uh, these developments in space sector are questioned for their sustainability, right? So what they want to do is they want to develop a launch vehicle which can be reused. And already they have tried to demonstrate some kind of technology for this, right? So you can go and have a look at videos, the videos up there, right? So reusable launch vehicle is another area where ISRO is working on right now. Okay, so that's mostly about the launch vehicles and the engines, right? That I want to talk about. Now let me just tell you a little bit of difference between a rocket and a jet, right? So let's try and understand. Let's try and understand. Um, yes, just give me a second. Yes. So let's try and understand what's a rocket and what's a jet. It's a very simple thing. Rocket and jet, right? So rocket is mostly used for upward motion. So wherever there's an engine and thrust is applied by that engine and the vehicle moves in upward direction, there's an upward thrust, and that is called as a rocket, right? But if there's a forward thrust, if there's a forward propulsion, that's called as a jet. Okay, I'm sorry, upward was called rocket, and yes, forward motion, forward thrust, that will be called as a jet, right? So uh, what happens is what happens is that mostly jets, they're not used for outer space activities. So if they're not used for outer space activities, then they can use oxygen from the atmosphere for combustion, right? Because there's oxygen in the atmosphere, right? So they can use that. But rockets, they cannot use oxygen from the atmosphere because they are going out of space, right? So there's no oxygen in outer space. This is why they need to carry oxidizer along with them, and we have seen that already. In these launch vehicles, there's always an oxidizer along with the fuel. But in jets, you can use oxygen from the atmosphere. So that's an advantage, advantage of some sort, at least for defense technology, right? You can develop supersonic jets and even hypersonic jets. So let's try and understand how these jets function. So initially, what happens is that you have a turbojet. So if you look at this, if you look at this diagram over here, you'll find that there are several gears, sort of gears or fans. They're mostly, they're called as fans. So what they do is these fans, they are rotated. When they're rotating at high speeds, they suck in the outer oxygen, right? Suck in the outer oxygen air, and then that air comes in and is compressed at very high temperature. Is compressed at high temperature, right? It becomes very, very hot. It becomes very, very hot. And when it becomes very hot, you add fuel to it, and then there is combustion of this fuel, and this helps in the movement of rockets, because then there'll be, um, if you've seen Iron Man, there's a fire coming out, and then he's able to fly, right? So the similar, you can imagine similar thing over here. There'll be uh, fire coming out, and then the rocket can move, sorry, the jet can move forward. So that is the turbojet, where air, you're sucking in air using the fans and compressing them at very high temperatures, and then combustion of fuel is taking place, and then jet is able to move forward, right? Now what happens in ramjet and scramjet? So you see, there were a lot of fans over here, a lot of moving parts in this turbojet, and this reduces the efficiency of machines. So if you know anything about machines, the more moving parts, the lesser the efficiency, right? So if there are more moving parts, then there's a greater chance that they'll be causing friction in the coming times, and they'll need a lot of maintenance. There'll be a lot of issues that will be there. So you cannot take these turbojets to very high speed, right? Because then these moving parts will start causing a lot of problem. They'll be causing drags. Cannot, it's just not feasible. So what happens is, you remove these moving parts in ramjet. In ramjet, what happens is that because there are no moving parts, but again, you need to have air coming in. So the jet is already moving at high speeds. When the jet is already moving at high speeds, what you can do is you can utilize that high speed to suck in air again, because the jet is already moving. So the air is already coming inside. Then again, you do the same process. You compress the air here, and you do combustion of fuels, and again, there'll be fire coming out, and the jet can again move forward. So again, that is the understanding for ramjet. And then an even better version of ramjet would be scramjets, which will be working at supersonic speed. So which will be sucking air at supersonic speed, and it will take your jet to hypersonic speeds. So when I'm saying supersonic, hypersonic, what does it all mean? Nothing much. It is just comparison with the speed of sound. So if you have a Mach number of 1, it means that you're moving exactly at the speed of sound. You are moving at the speed of sound. If your Mach number is 2, it means you are moving at twice the speed of sound, right? And if you are between Mach 1 and 5, then you will say that you're moving at supersonic speeds. And if you get in 5, then you'll say that you will moving at hypersonic speeds, right? And below 1 definitely will mean subsonic speeds, which we are usually accustomed with. But if you look at supersonic and hypersonic jets, these are very, very fast. So you see, they're moving faster than speed. I mean, faster than the speed of sound. I mean, that's really, really fast. So supersonic and hypersonic is where we usually pay most attention to, because they are more quick. And then if you want to design a missile system, you want to be quick, right? You don't want your missile to be flying at a very low speed, because then there's a greater chance of it being uh, neutralized. Okay, uh, so you can see that there are three stages. One is compression, then there's combustion, and then there's an exhaust. In all these three, there is the basic thing that's there. And you can see, as the design goes, the moving parts and other things, it keeps on getting uh, lesser and lesser, and the jet becomes more and more efficient to move at higher and higher speeds, right? So let's try and have a look at the basic differences. If you look at turbojet, the air intake will be happening at subsonics. In ramjet, it will be at supersonic, and in scramjet, it will be at hypersonic. And if you look at moving parts in turbojet, there will be moving parts. In ramjet, there will not be. Scramjet, there will not be any moving parts. Speed range, turbojet will fly at subsonic to low supersonic, right? So not more than that, because I told you moving parts, so efficiency will not be there. Ramjets can move up to 5. At 5 or 6, they'll start again cause tracks. So again, That is why we need scramjet, which will move at hypersonic speeds. In combustion, here it will happen at subsonic, here it will happen at subsonic levels, and here it will happen at supersonic levels. All right. Um, Again, efficiency, it will be high at subsonic speeds. Here it will be at supersonic speeds. Here it will be at hypersonic speeds. Right. And the usage, you can look at the usage on your own. Commercial military uses are mostly the most common ones for all kinds of jet engines. Right. So again, for space technology in particular, they're again trying to use scramjet in some way. They're trying to utilize this engine for at least the part when you are when the rocket is in the atmosphere. When you're still in the atmosphere, you have the advantage of oxygen, right? So if you're using scramjets, again, you can move at very high speeds, hypersonic speeds, and that can help in the liftoff of the rocket, of the launch vehicle, right? So at least for that purpose, they're trying to use scramjets, right? So that's mostly about the different types of jet engines, right? And yes, the differences are there for you to see, and yes, that's the difference. Okay, so now we've covered the basic concepts of launch vehicles, engines, uh, rockets, and jets. Okay, and now I want to talk about the outer space in general, right? So I'm not going to talk about specific things like moons or planets. I'm more interested in the general overview of how things actually um, come up in space. You know, what is the basic element formation that is happening outside, right? And once you have an understanding of this, maybe you'll have a better understanding of everything else also. And usually a lot of questions come every 2 years, 3 years. We see a question from these concepts. Last year itself, there was a question from this concept. I remember there were two statements, right? And there's something about a dwarf star. I know if I correctly remember. There were two statements. One was a statement, and another was a conclusion, and then that you have to answer. So anyway, these are important concepts nonetheless, and let's try and dissect what happens when elements are being formed in the universe, right? So usually what happens is that you have a stellar nebula at the beginning. Stellar nebula is just a cloud of dust, gas, and other particles in space, right? And when this nebula comes together, right? There'll be gases which will come together, and either it can form a small average star or a massive star, right? Now, when it does that, in both these cases, what will happen is that there'll be nuclear fusion reactions that will be taking place. Nuclear fusion reactions that will be taking place because there is hydrogen definitely. So there will be nuclear fusions happening, and then these reactions will continue. Will keep on continuing for a long period of time, and eventually, what will happen is that if your fuels if your fuel is hydrogen, initially it's forming helium, and slowly it will start uh, becoming heavier and heavier because you'll run out of hydrogen, but then you'll obviously have some helium, some hydrogen. They'll combine and form other heavier elements like carbon, oxygen, nitrogen, right? They can go up till iron. Similarly, here they keep on combining. I mean, they keep on forming heavier and heavier elements. So now you're trying to understand every element is being formed inside the star. Everything that you see around you was once part of some star. That element, whatever it is made of, that element is was made in star. So when it keeps on becoming heavier and heavier, this is where we reach the stage of red giant or super red giant. And when it becomes so heavy that cannot hold its mass on its own, it explodes. Right. So if it's a red giant that causes a planetary nebula, and then there'll be um, white dwarf left at the center, which is a very, very dense object, and rest of the materials will be thrown out. So there'll be planets being formed from this, and other space objects, and there'll be a white dwarf left at the um, center, which is a very, very dense object. And in case of superred giant, there will be supernova explosion. Now, this supernova explosion will result in either a black hole, or a neutron star. Now, a black hole is again uh, something which has a very, very high gravity, such high gravity that even light cannot escape. So either you will have a black hole or a neutron star. Neutron star is again a very, very dense object, very, very dense object. A spoonful of neutron star can have the weight of 100 suns or 1,000 suns. So it's very, very dense, right? It's extremely dense because neutron. It's called a neutron star because neutrons are very, very tightly packed. Even protons combine and they start making neutrons. So everything inside is essentially a neutron, tightly packed. There's really no space inside. So that is why it's extremely dense, and it's so dense. There's so much of magnetic fields around this, right? And sometimes there'll be electromagnetic radiations coming out, and because sometimes they're spinning, so it will look like a pulsating light. So at that stage, that will be called as a pulsar. There are other types of neutron stars that you can have a look at, right? So different types of neutron stars are there, but again, one of the most dense objects known to us, right? And then the black holes. Black holes, I think a lot of um, you will already be familiar with concept of a black hole, um, which is essentially anything that reaches a black hole will be taken in, right? Because of its high gravity, right? And they're also very one of the most mysterious objects. A lot of research is being done in their understanding, right? And all these giant objects, usually when they are so let me give you a little bit of perspective on gravitational waves also. So these are very, very highly mm, uh, dense objects. You can see very, very heavy objects. And when they are moving around space, uh, because of their massive gravity, uh, they cause ripples in space-time fabric. So if you have a space-time fabric, something like this, right? Something like this. And suppose I put a very, very heavy element on this fabric, you will find that there's uh, there is there is a depression that has been caused, right? There's a depression that has been caused. This depression that has been caused, and if they're moving these objects, if they're moving at a high speed, then they cause ripples in these in these fabric in this fabric, and these ripples they are actually called as the gravitational waves. All right, so you can have a more of a more you can have more uh, of a deeper understanding by reading more about it. But this is essentially the concept. Very, very massive objects with high gravity moving at high speeds, they can cause these gravitational waves, which are noticeable. I mean, everything can cause gravitational waves because everything has uh, a gravitational effect. But again, uh, noticeable effect can only be caused by things like black holes or neutron stars. This is why when they're designing those missions, they're studying these black holes for detection of gravitational waves, right? So this is why these basic concepts are important because now when they are developing those missions of LEGO, or what are they actually, why are they actually going for these things, right? So this is the reason why concepts are extremely important, right? So that is the mostly about the space tech. Uh, Now we'll come to the biotechnology part of the. All right, so let's now look at some basic concepts related to biotechnology. And if you want to read biotechnology, I think NCRTs, they cover a lot of concepts related to this particular topic. If you look at other topics, maybe you'll find that there's some content that you won't be able to exactly locate. But as far as biotech is concerned, especially 11th, 12th NCRTs, they are very, very good. Okay, so what I'll try to do is I'll try to go about some very basic things that are required from the perspective of uh, bigger concepts that will come in the future series, in the first future sessions that we'll be doing in this series. Okay, so for that, I want to just build up a little bit of base. So I'll do something related to nucleus, related to chromosomes, DNA, RNA, and gene editing, right? And then we can look at the other applications that will be there in the news items, or other deeper concepts. Okay, so let me just go straight to nucleus. Okay, so if you want to refer to you know animal cells, plant cells, you can go and look at the NCRTs, right? So again, when you're reading about cells, there'll be different organelles, right? There'll be different organelles, and they'll have their own functions, right? So even those are important, and you can go and have a look at it because there are so many of them, and we don't have time to cover it in this particular session. I'd recommend that you have some kind of familiarity, and it's all just logic, because cell is also a living entity of its own. It will have a brain, which we are going to study right now, the nucleus. It will have something to give energy to the cell. It will have something that will be throwing out wastes, something that will be transporting substances, right? So all these things will have a name, right? And those are the organelles. Okay, so one of those organelles is the nucleus, right? It's nucleus. And nucleus is one of the most important parts of a cell because it dictates what the cell has to do, right? So all the information, the relevant information is present in the nucleus, and it's present in the form of DNA, right? It's present in the form of DNA. But before we get to that, let me try and go one by one about different parts of a nucleus. So because, as I said, it's a very, very important part of the cell. Definitely, it will have some protective coverings. Those are called as nuclear envelope or nuclear lamina, right? And then there's a liquid like substance present inside the nucleus called nucleoplasm, right? And then uh, there is something called chromatin fibers. We'll come to that. There's something called chromatin fibers. Just understand this for the while as some kind of thread-like thing that's there inside the nucleus, right? And then at the very center, you have a nucleolus. Now, nucleolus is the place from where ribosomes will originate. It's the place from where the ribosomes will originate, and they will exit the nucleus through these nuclear pores. Right. So because if you know about ribosomes, they're also one of the organelles, cell organelles, and they are the site of protein synthesis. So most of the times, what is the cell doing? It's making proteins, and ribosomes are the ones which are helping in this process, right? So in the making of proteins, they play a very important role, and they come from the nucleolus, which is all, located at the center of the cell, right? So if you remember, I said I'll come back to chromatin fibers. Now, chromatin fibers, they are just thread-like things, but it's actually all your DNA, right? It's actually your DNA. So let's try and look at it. So this is how your chromatin fiber usually looks like when it's present inside the cell. Okay, chromatin fibers. All right. Now what happens is this this has to condense into a shape. This has to condense into a shape when the cell has to undergo division. Right, this has to condense into a shape, and that shape, when it condenses into a shape, it's called as a chromosome, right? And what is all this? I'm just dropping names right now, but what is all this actually? In reality, if you look at this diagram, when this um, chromatin fiber, it condenses into a chromosome, right? Again, so if you pluck a thread from here, what will it be? It will just be the fiber itself, right? It will just be the chromatin fiber. So you have this chromatin fiber all along, and if you look deeper and deeper into it, you'll find that there is some sequence, there's some repeated sequence. So nucleosome is a repeated sequence of something, and that something is DNA is DNA, which is wrapped in histone proteins, right? Which is wrapped in histone proteins, right? there are specific numbers as to know there's how many strands of DNA are wrapped around how many uh, blocks of protein. We don't need to go into that much of detail. But remember that DNA is wrapped around these proteins in a sequence and in a shape called a double helix. Right? So this spiraling ladder-like shape is called as a double helix, and this is how your chromatin fibers look like in reality, right? So what happens is that during cell division, because the complete genetic information needs to pass on to the next generation, the cell cannot leave its genetic material like this, and you can't divide it like this, right? The cell cannot divide like this, so it has to condense into a proper symmetrical shape like this, right? So that the division is possible. And if you want to know more about how cell division happens, you have to again go back to your NCRTs and read about mitosis and meiosis. Okay, so these are different types of cell division because there are different types of cells in your body. One is the somatic or the body cells, and then there'll be sex cells. So all of these have different processes. Please, if you want to go into a little bit of depth, please read about mitosis and meiosis as well. Okay, so I just want to, for the moment, I just want you to understand that there's something called chromatin fibers, and that's nothing but just your DNA wrapped in histones, you know, and when it condenses into a shape, it's called as a chromosome. Okay, so that much of understanding I want at the moment. Okay, so if this is understood, let's move on. Let's move on and go a little bit deeper into the DNA. I told you it's a double helix shaped structure, and if you look at it, if you try to straighten it out a little bit, you'll find that there's a sugar phosphate backbone to it, right? There's sugar phosphate backbone, and there is nitrogenous bases. These are nitrogenous bases. So there is four of them: A, T, C, and G. These are your nitrogenous bases. So adenine, thymine, cytosine, and guanine. Okay. Okay. So these together, if you look at this sugar phosphate backbone, right? If you so just magnifying further into it, so you look at this sugar phosphate backbone along with this nitrogenous base together, it will be called as a nucleotide. All right, so together it will be called as a nucleotide. Okay, and and if we don't count this phosphate backbone, if we just look at the base and the sugar, if we just look at the base and the sugar, then it will be called as a nucleoside. It will be called as a nucleoside. All right. The base and the sugar together will be called as a nucleoside. Okay, so if these terminologies are clear, let's move on to a slightly more interesting concept, which is genes. Okay, it's genes. So I told you that there is a cell. Inside the cell, there is a nucleus. Inside the nucleus, you have DNA. Okay, uh, of course, you know that it's in the form of chromatin fibers, and when the cell has to go division, it will become chromosomes, right? All that you know. But there is DNA. Essentially, there is DNA inside the cell. Now, if you look at this, okay, if you look at this DNA, suppose let me take a very specific example for your eyes. Okay, so let's take the example of blue eyes, right? Let's for a moment, let's say that this is a cell which gives the eyes blue color. It's responsible for producing a protein, right? It's responsible for producing a protein that gives the eyes their blue color. Okay. Okay. So how will it do that? How will it do that? So uh, if you look at this, if you look at this structure of DNA, what you find that what you find that is all the A's are bonded with T's in a double bond, and all the C's are bonded with G's in a triple bond. All right. And this is constant. So, you can't have an A bonded with G or a C bonded with T, right? So, when you have something like this, can I say, if I write A, C, T, G, something on one side, right? Can you tell me the other side on your own? Yes, you can. A is bonded with T, C is always bonded with G, T will be bonded with A, G will be bonded with C, right? And so on and so forth. So I just need to know one side. I just need to know one side, and the right side will always be known to us, right? Because we don't need to figure out both of them together. No, if we know one side, we will know what is going to be on the other side because of this rule. Okay. Okay, so let's come back to this, and let's say that this DNA, I'm going to label it. So let me say that it's labeled as TACGCG. Okay, something like this. So if you have this sequence, I can go on and say it is CAA something. Okay, let's assume that I've taken a DNA and one side of the DNA has these kind of nitrogenous bases. Okay, now I am saying that this DNA is present in this cell, which is going to be producing a protein for blue eyes. So where is this information stored? Now that is your genes, right? So this, if I say that this particular um, combination CGC is the code, let's for a moment suppose that it's a code that when the cell reads this code, this particular cell, it reads this code, and it knows that it has to produce a protein that will give the eyes its blue color, and that will be called as your gene. That will be called as your genes, right? So these are coding genes. So which means that they have certain they have certain traits, right? So those are your genes. They have a specific function, right? So what happens is in your eyes, now you should have a valid question. What about the other genes? You know, there could be a gene for giving blue color, brown color to your hair. There could be a gene for digestive enzymes. There could be genes for so many other things. There could be genes for, because there's so many proteins in our body. There could be genes for so many other things. Then what happens to those genes? Because inside the DNA, inside the nucleus, the DNA is going to be complete, right? So you'll have all the information, right? So why are we only saying uh, that this cell is going to produce blue color for the eyes? Because this cell knows that it has to do that, and this gene in this cell will be turned on and the others will be turned off. Similarly, if I take the example of your stomach, the required protein that your stomach cell has to make, it will make only that protein, and the rest of the proteins, including this one, would be switched off, right? So that's how it works, right? That's how it works. And this concept is going to be important when you come to gene editing, right? That is how we identify and go to a specific site and try to edit that gene. Okay. Okay, so I hope this is clear. I hope this is okay with everyone. And now if you look at some other terms which are related to these things. So we studied about a single chromosome. Now when the cell will be undergoing cell division, it will duplicate, the single chromosome will duplicate itself, and this will be looking like this, in a form of duplicated chromosome, right? So these arms of the chromosomes will be called as chromatids. Its center will be called as centromere. There will be a short arm, there will be a long arm, and these chromatids, because they've come from the same chromosome, it will be called as a sister chromatid. All right, so these are just simple terms. Now let's come to the understanding of autosomes and sex chromosomes. So in humans, what happens is, till now I have just shown you that okay, there is a chromosome, there's something called chromosomes. But in humans, there are 23 such pairs of chromosomes, right? So there are 23 pairs of chromosomes, 23 pairs of chromosomes, right? So the first 22 are related to your body. They give the body your general body characters. And the last one, the 23rd pair is the sex chromosome. So it determines whether you're going to be a male or female, right? So that is the function of that 23rd chromosome. Okay, so 23rd pair. Okay. All right. So if I want to tell you that if you have a sequence like this, 1, 2, 3 up till 22, and then you'll have X. This could be one thing, right? Um, and X could be paired with X, or then if you have something like this, it have an X paired with Y. So X and XY, no, I'm just giving names right now to the 23rd pair, but I tell you what it means. Okay, so if you have a sequence which is ending in X, and then you have a sequence which is ending in Y. Okay, so this is the sperm cell. So if you notice, so I've just given one single line to it. So this means that these are not pairs. Now, so your sperm cell will have 23 chromosomes and not 23 pairs. Similarly, your egg cell will have 23 chromosomes and not uh, 23 pairs. Why? Because see, in your body, in your body in totality, you have 23 pairs. That means it is 46, right? So your eggs and sperms, they have to combine together, right? They have to combine together. So the first 22, they will definitely combine, right? That is how you're getting these pairs, right? One from mother, one from father, right? So that is how they'll combine, right? That's the story of first 22. And the last one. So let's try and understand how chromosomes actually look like. Um, I'm not going to see them under some microscope, but if you look at the figure and you look at there are two kinds of chromosomes that are broadly classified in this figure. So what is the autosomes? So autosomes are the ones which are related to your bodily traits, and then is the sex chromosomes which define whether you are a male or a female. All right, so uh, before that, let me just give a passing mention to diploid and haploid cells. Diploid and haploid. Okay, so cells uh, which have n number of chromosomes, they are haploid, and if they have two n number of chromosomes, they are called as diploid. This is I'm making it very very simple. Um, Now, what do I mean by this? Let's take the example of human body itself. In humans, in total, we have 46 chromosomes, right? So 46. So we have 2 into 23, or you can say 23 pairs of chromosomes. So that right now that you see over here, there are 23 pairs, 22 plus this, the last one. Okay, so the last one is the 23rd pair. So you have in total 23 pairs of chromosomes, right? Or in total 46 chromosomes. So I count 1, 2, 3, 4, and I can count last 45, 46. All right. So that's what your diploid cell is. And then you have something which is called a haploid cell, which has only n number. So it is 23. So it will have, for humans, I'm talking about human body, it will have 23 chromosomes. Okay. So what happens is that normally all your body cells have the entire genetic information that is required, that is required for your body. So you take any body cell, skin cell, stomach cell, eye cell, anything, All of them will have inside the cell there will be nucleus. Inside the nucleus there will be 46 chromosomes in total, like the ones you see here, right? So out of the 46, so these 46 will be occurring in pairs, right? Like you can see 1 2 3. So there will be one pair, two pair, three pair, right? And the last pair could be XX if you are a female. So this is why this is the 23rd pair, right? This is the 23rd pair. Or it could be XY if you are a male, right? It could be XY. Okay. All right. So what happens is that your um, sex cells, which is your eggs and sperms, are actually haploid cells. So why? Because you have n number of chromosomes over here and n number of chromosomes over here, and when they both come together, that is when you will have in total 2n chromosomes, which is the actual number of chromosomes that you need to have, right? So you can't have 2n in eggs and 2n in sperms, then in total you'll have 4n. So then you're not human anymore, right? So whatever you are, you're not a human. 46 chromosomes make you a human. You cannot have 108 chromosomes and still call yourself a human, right? So that is why the sex cells need to be haploid. And when they are haploid, it means that they have 23 chromosomes in total. Now, listen carefully. If they have 23 chromosomes in total, all right, and I just said that in a human cell, body cell, 23rd pair is either XX or XY, right? So here, let me just try and no, this one two over here is the pairing, right? So these are pairs, all right? Okay. So let's say here you have um, a haploid cell, or let me do one thing. Let me just draw it once again for you. So if you have these are 23 chromosomes, right? So the last one could be X, or it could be Y, right? Okay. So when you are a female, you will have XX, and when you are a male, you will have XY. Okay. Now listen carefully. You know, what happens is that when fertilization takes place, right? So let's suppose here I'm saying that these are coming from your female, these are coming from your male, right? This is what can happen, right? Because only males can carry this Y uh, chromosome, right? Okay. So now what happens is this I'm saying is one XL, and this again is another XL. This is a sperm, right? And this is again a sperm. Okay. Just for your understanding, I'm trying to go a little bit of deep into the concept. It's actually pretty simple after this. So what happens after this? No, when male and female cells, sex cells, they will combine. What will happen? Either you can go for this combination X with X, right? Or you can go for this combination again. It will be X with X, right? Or you can go for this combination X with Y, and this again X with Y, right? So what do you end up with? You end up with two possibilities of a male child, sorry, of a female child, and two possibilities of a male child, right? So this is how it happens, Obviously other pairs will also combine. That's how you'll have, you know, these these will also combine together. No, eggs, not eggs and eggs, but you get my point. Let me, I think I hope I've made my point clear. This will combine with this will combine with this, and so on and so forth. Right. So this is how you define diploid and haploid cells. So your sex cells will mostly be haploid cells, right? And in general, all your body cells are diploid cells, right? So that's what you need to know. And that the 23rd pair actually determines uh, whether you are a male or a female, right? So if it's an XX, it's a female. If it's an XY, it's a male, right? So let's move forward. Let's move forward, and let's quickly glance. Take a glance at the differences. Autosomal chromosomes means soma means body. So these are body chromosomes. They code for traits that do not depend on your gender. So these are things like hair, eye color, ear shape, right? And then there are sex chromosomes, which make a person male or a female, and they code for specific gender traits like baldness, more found in males, hemophilia, color blindness, again more found in males, right? So um, these are the essential differences that you will find in the two types of chromosomes. Okay, so let's try and you know look at DNA and RNA. So you have done the structure of the nucleus. You've gone inside the nucleus, have a look at the chromatin fibers, and we understood what chromosomes are, the body chromosomes, sex chromosomes. We understood uh, the DNA, how it looks like. There's something called an RNA also, which is a single stranded, you can call it a single stranded DNA, but it's actually a single stranded um, chain of nucleotides. All right, so let's try and understand this. Let's try and understand this table by looking at the differences. All right, so your DNA is also called as the deoxyribonucleic acid, and RNA is called as the ribonucleic acid. All right. So DNA, it has four bases: adenine, guanine, thymine, and cytosine. We've already done that, and it has a deoxyribose. That is why it's called as deoxyribose nucleic acid, and a phosphate backbone. We have seen the phosphate backbone. This is the name of the sugar, right? This is the name of the sugar, right? Right? So if there's deoxyribose over here, in RNA there will be a ribose sugar and a phosphate-based polymer having again four different bases. But note that instead of thymine, you have uracil. So you have adenine, uracil, guanine, and cytosine. Right? So you have something called AUCG. Here you have ATCG. Okay. Right. Now, a DNA is found inside the cell's nucleus, and some of it is there in the mitochondria also. It's very important to know that some of the DNA exists, around 0.1% exists in the mitochondria also, which is the reason for mitochondrial diseases, right? There's something called mitochondrial diseases, right? So DNA is also found over there. But if you look at the RNA, the RNA is found in basically three different places. One is the nucleus, right? Then there is ribosome, and then there is cytoplasm, right? So in the nucleus you have what is called as mRNAs or messenger RNAs. So you might have heard of mRNA vaccines. So the mRNAs lie inside the nucleus. Then there's something called rRNAs, which are there in the ribosomes, and there's something called tRNAs, which are there in the cytoplasm. Now what are the different functions? We'll maybe if we have time we'll look at it later, right? But that's basically the three types of places where RNAs would be found, and in three different forms. Okay, so the sugar we have already seen. Here it's deoxyribose, and here it is ribose, right? Now what is the function? The function of DNA will be to transfer the genetic information, to make it possible to transfer genetic information. So when the cell divides, basically what is happening is the genetic information has to be passed, right? So DNA is the one carrying that information. All right, so it's a long-term storage medium of that information. And what about RNAs? The RNAs Transmission of the genetic code required for protein production from the nucleus to ribosome is accomplished by RNA. Now, there's a whole process of very, very complex, extremely sophisticated chain of activities that happen before a protein is formed, and those activities happen through these single-stranded nucleotides called RNAs, right? Um, Now the whole process, it will take a little bit of time if I go deep into it. Um, but understand it in a very simple way. What happens is that from the nucleus, the message will come, right? Then the message will reach the site of for protein synthesis, which is the ribosomes, and then there will be a chain of amino acids that will be combined together, give shape to the protein which the cell has to make. It could be a protein for digestion, it could be a protein for your blue eyes or brown hair or anything, right? So that's how it happens. Okay. Now in DNA, the predominant structure is the double-stranded molecule, a long chain of nucleotides, right? Here it's a single-stranded molecule and a shorter nucleotide chain than DNA. All right. Then DNA replicates on its own. It's a self-replicating thing. RNA does not have the ability to reproduce on its own, right? Whenever it's needed, it's made from the DNA only. Okay, it's made from the DNA only, right? And the pairing we have already seen, A combines with T, right? With a double bond, G combines with C. In a triple bond, here it's the same thing. It's just that A combines with uracil, and G remains the same, combining with C. Okay, so these are some of the essential differences that you need to know between DNA and RNA, right? Okay, so let's come to CRISPR-Cas9. Now we have looked at the nucleus, we've looked at the chromatin fibers, we've looked at the DNA, the genes, right? And we've looked at the autosomal chromosomes and the sex chromosomes. Now, a very important thing to understand here is that there will be times when you will find that gene editing is going to help you out. So there are things like gene therapy. There's a whole branch of study called gene therapy, which is focused on finding health solutions by editing genes, right? So eventually you'll find that gene editing is something that is required for these kind of purposes. And then what do you do for that? There's a lot of complexity attached to the genes itself. What do you do to edit them, right? So that field of study is called as genetic engineering, and one of the foremost techniques that are currently in use, most versatile. It's a little bit expensive, but it's very, very accurate, very, very precise, and that's called as a CRISPR-Cas9 technique. So CRISPR-Cas9 is basically going to edit your genes, right, at the required site. So before I explain this, just remember that whenever I say nuclease, it means molecular scissor, right? So nuclease is actually a type of enzyme. Enzymes, they're a type of proteins that facilitate biological reactions. So here it's the same thing. It's an enzyme which is going to facilitate this reaction. It's actually a molecular scissor. It's just going to behave like a scissor, right? And we'll understand what it actually is doing. So your Cas9 is actually the full form of Cas9 is CRISPR associated nuclease, right? CRISPR associated nuclease. Okay. Now it's actually a little bit more complicated than I would explain it. I'll try to simplify it or oversimplify it for you. But in reality, the processes are very, very complicated, right? So, but for our understanding, this much is enough. Uh, the diagram that you're seeing here. So what actually is happening is that if you want to edit a gene, you need a location, right? So uh, you will have a guide RNA that is going to guide you to the location where you want the gene to be edited, where you want to edit the gene, right? So how is it going to do that? So, no, you can see that it's just a single strand of nucleotide. So what will happen? It will have some kind of patterns A, T, C, G, G. something. Now, this guide RNA, along with Cas9, together is forming a complex over here. So what happens is that you mix these two or you send them to together to the site, right? So guide RNA along with Cas9 which will reach this site. What is this site? It's the opposite of the one, the code that is being carried by this guide RNA, right? So it will identify this location wherever ATCG could pair, right? It will identify this location, and once the location is identified, once it will act like it has found its matching parts, and once it finds the exact location, the Cas9 enzyme will begin to do its work. You know what the work of a nucleus would be? It would be to cut, right? Because it's a molecular scissor. So it will go and cut the gene at this location, and when that happens, your gene looks something like this, and then you can do anything you want. You can either insert another DNA segment, another gene to it, or you could leave it like that. If you leave it like that, the natural process will take over. The DNA will repair itself, right? Or you can do anything else that you want. If you want to duplicate something, you want to add a new uh, segment, up to you, right? So this is how CRISPR-Cas9 works. It's fairly simple. Uh, Guide RNA along with Cas9 will go to the location where the gene has to be edited. It will cut the gene. The Cas9 will cut the gene at that location, and then you can do all sorts of things that you want with it, right? So that's your CRISPR-Cas9, and when we deal with news items and a little bit of deeper concepts, then we'll try and understand what is actually the applications of all these types of technologies. Okay, so that might happen in the future sessions. But for right now, these are the concepts mostly broadly the ones that are associated with uh, biotechnology, right? Okay, so let's go to a slightly different topic, but it follows from biotech itself. Now we're trying to understand the different types of microorganisms, right? And what are their characteristics? Right. So uh, bacteria, viruses, and fungi, these are mostly the ones that we want to look at, right? Because most of the things that are asked in your exam, they are centered around these three kinds of microorganisms. Of course, there are more, but we are mostly trying to, at least at this stage, we'll mostly looking at these three. All right, uh, so let's try and understand what happens in these three things. So if you look at bacteria and fungi, uh, there's no doubt that they are living beings, right? But if you look at the virus is actually, if you define it in a technical way, it is an obligate intracellular parasite, right? And what that means is that it cannot live on its own. It needs a host cell, right? It needs a living host cell. It cannot live by its own, right? And virus is always a harmful thing, right? So virus is always considered harmful, all right? So let's try and go one by one. If you just, I just wanted to give a little bit of this minute difference that's there between virus and other microorganisms. Virus is a parasite, right? It cannot live on its own. So if you are with me on this, let's go one by one and look at each of these differences. Now, bacteria obviously need a microscope to see. Viruses, they're smaller than bacteria. Again, you need some kind of microscope. Fungi, what will happen is single cells. You need to see you need a microscope. But there are also multicellular cellular fungi like mushrooms, right? So they are all around you. You must have seen those things, right? So uh, that's the first thing. Then bacteria would need warmth, moisture, and nutrients like other. other microorganisms which are living. Then fungi will also need warmth, moisture, and nutrients. But viruses, I said, they depend on living hosts, right? They're entirely dependent on the living hosts. Then if you look at bacteria, they're divided into aerobes and anaerobes. Again, aerobes means they'll be able to survive in the presence of oxygen only. Anaerobes will mean that even in the absence of oxygen, they will be able to survive, right? Similarly, the same classification holds for fungi as well. And viruses, as I said, they're on the edge of life. They are not living until they find a living host, and they are not classified into any such kind of categories. All right. So viruses are always parasites. Now, um, bacteria and fungi could be saprophytes or parasites. What's the difference? Parasites, they will either live on or inside another organism, right? While a saprophyte is something that will feed on dead and decaying matter, right? So that is the difference. That is something that you need to have a little bit of clarity on. Most of the times people will get confused. But saprophytes, they feed on dead and decaying matter, but um, parasites are usually going to live on or inside another living organism, right? So viruses are always parasites, okay, and they're always harmful, right? While on the other hand, bacteria could be beneficial or could be harmful. There's bacteria in your gut, which is helping in the digestion, right? The same holds for fungi. It can be beneficial, it can be harmful. Right? There's some fungi that you can eat, right? So it's beneficial that way. But there are others that can cause diseases which are poisonous, right? Right. So yeast is an example of bacteria that is some way beneficial for us. Okay, right. So you can read these other points like what kind of diseases are they causing and what are the uses. So bacteria can be used for making antibiotics, or it can be used for fermentation. Same thing holds for fungi. It can be eaten, can be used for making beer, bread, antibiotics. All these are different applications of these microorganisms. All right. So that's a basic difference in these three. And it's not like these simple concepts are just because these are very simple things they can be ignored. There was a question um, a few years ago on, I think 2 or 3 years ago, on fungi. And a lot of it could be answered using this slide, right? So it was about different types of mushrooms, right? And if you had some clarity on the characteristics of fungi, you would be able to answer it very easily. It was based actually on a news item in Northeast. So that is why these concepts, always these concepts will play a very important role when you are going for your prelims or even in mains. Okay, so that's the difference between bacteria, virus, and fungi. Now, because you have mentioned that all these things, microorganisms, they can cause diseases, or they can cause diseases also. And when they cause diseases, they'll be called as pathogens. Pathogens, all right? They'll be called as pathogens. Now, our body has a system to fight these diseases, right? That's called as the immune system. There's the disease resistance system that is present in our body on our own, right? So there's some kind of immunity that is inherent in us, and there is some kind that we can acquire, right? So I just want you to understand at this stage what happens when you're trying to acquire immunity using things like a vaccine, right? So there could be several types of vaccine. There could be live attenuated vaccines. There could be um, pneumococcal vaccines. There could be mRNA, could be oral, nasal, all sorts of vaccines are there. Um, right. So all these vaccines, even though there are different types, or they could be protein subunit, but even though there are different types, all of them are essentially working on the same concept, which is to give a immunologic memory to your body, right? So what happens is, we look at this graph. We look at this graph. Okay. Okay. So what happens is that the first time you encounter what do you call any kind of infection, right? Your body will respond to that, right? So there's actually different layers of your immune system, or you could say that there are different layers of your defense system, right? Which will be trying to fight that foreign substance or any kind of infection. Okay. So what happens is in the very first exposure, your body is not prepared to handle it, right? In the very first exposure, your body is not prepared to handle it, right? So that's called as the primary response when your body is not prepared to handle it, and that is when you actually get the disease. Okay. This is the primary response. But what happens is after this primary response, your body develops an immunologic memory, right? Your body develops an immunologic memory. And so, if the same virus or same bacteria enters your body once again, then your body will have a secondary response. And the secondary response is going to be much more efficient. Why? Because now you have a memory of the previous invasion by the virus or bacteria. So now what you can do is your cells, they know how to deal with them, right? So they know how to fight with them. So now they can come in great numbers, and because they know who to kill, when to kill, where to kill, your body will have a much better response, and that will be called as a secondary response. So this is the principle that is used by vaccines. Vaccines will try and give you a little bit of um, a weakened form of the infection. Since in some cases they will not even give that give that infection. But leave that aside. When we discuss types of vaccine maybe later, then we'll come to that. But for now, just understand this in a manner that the vaccine is trying to actually drive the primary response, so that when the actual, so in the vaccines, most of the times you will not have the actual virus or bacteria that is causing the disease, but you will have a weakened form of them or a weakened protein subunit of them, a weakened antigen, anything else you will have, or an mRNA, right? So you'll have different, you could have any other thing, but you'll not have the real virus. So what will the vaccine do? It will train your body to develop that primary response, so that when the actual disease or actual virus or bacteria tries to enter, then your body produces a much better response, right? That is the secondary response. All right. So that's about the immune system and what the principles that the vaccines will use. Right. Let's move forward and try and understand something called antimicrobial resistance. Right. Antimicrobial resistance. So we looked at bacteria, viruses, and fungi. And I talked about now how viruses are always harmful. Bacteria and fungi could also be harmful. Uh, so let's take the example of a harmful bacteria. Right. Right. What happens? See, bacteria has been here on Earth for billions of years, much before we came in, right? So they definitely have evolved a lot over the period of time, right? And they have been very, very successful living beings. So that makes them, in a way, very unique. But for us, it also creates a certain level of headache, because what happens is that if you have a harmful bacteria, I'm taking the example of bacteria. If you have a harmful bacteria, for this harmful bacteria, what you'll do is you'll take an antibiotic, right? You'll take an antibiotic. All right. So what happens is this antibiotic enters the population, right? Because a lot of people are taking it, and this antibiotic is trying to kill the bacteria. But if you remember those ads from your childhood days, that there's some bacteria that's always left. You can only kill 99% of the germs, 99.9%, but there'll be some bacteria which will not be affected by this antibiotic, right? Right. So you can see in this diagram that a lot of germs are there, but very few are drug resistant. So antibiotics, they kill almost all bacteria except the drug resistant ones. So the, you know,
If there are so many over it, maybe 1%, 0.1% will be left, right? They'll not be affected by the antibiotic. But eventually, what will happen is that these antibiotics will not be able to overpower these drug resistant bacteria, because what happens is that the bacteria will realize that this this form this form is beneficial. If you remain in this form, then this antibiotic can do nothing, right? When it realizes that, it starts to grow and take over, right? So this drug resistant bacteria, it grows and takes over. And when that happens, it's a problem for us, because now antibiotics are not going to solve your problem, right? Now you'll have to develop uh, another stronger antibiotic to deal with these bacteria, drug resistant bacteria, right? And then again they'll become resistant, and you'll have to make an even stronger antibiotic. So now this, antibiotics, if you keep on making stronger and stronger antibiotics, they also be having side effects, right? So that's going to cause a lot of problem. And every year, millions of deaths happen throughout the world because of this reason, because of antimicrobial resistance that has been developed over the years, right? Right. Right. So this is what actually happens. The drug resistance keeps on getting transferred, and it spreads throughout the population. So drug resistant bacteria become widespread, and then your antibiotics stop being effective. Right. So this is a major concern, and WHO has a whole program for this. The government has a whole program for this. Right. So this is why we are studying all this. So whatever we are studying, the reason we are studying is that because there's some policy implications of all these things. There's some um, scheme running, there's some campaign running, some program running behind all these things, uh, to uh, to save the lives of people, or to benefit the population in general, right? So this is why we need to know these concepts, right? So AMR, every year you'll find articles, every few months I find articles in the Hindu about how AMR is impacting the world. So how is it affecting the economy, right? And why no one is talking about it. So all these kind of articles you'll find in the newspapers, read about it, and will get a better understanding of what's the impact of all these things. Okay, so that's antimicrobial resistance.
Okay, so let's now try and understand something related to nanotechnology. So we've covered space and biotech and health um, quite fairly, and now we'd like to look at something called materials at nanoscale. Right. So why is nanoscale important? It's because when you try and manipulate materials at very small scales, these are atomic and molecular levels I'm talking about, right? At these levels, when you're manipulating materials, they start showing different properties, right? So your gold, usually it's the color that you see, your yellowish or golden color. Uh, if you have gold nanoparticles, they could be red, it could be black, it could be any other thing, right? So uh, it's just an example of physical property that I'm telling you about. But then there is electrical properties that can change. So carbon, if you have carbon nanomaterial like graphene, right? Now that is a very good conductor. It's very strong. Uh, but graphite, graphite is the carbon that you see, uh, like they usually you see it around. That is very brittle. So properties of materials start to show differences, significant differences, when you start manipulating them at atomic and molecular levels, and that is the magic of nanotechnology, right? So there was a physicist by the name of Richard Feynman. In 1965, he had said there's plenty of room at the bottom. So what he was referring to was the empty space in the atom, right? So there's a lot of empty space, more than 90% of it is just empty, right? So if you are able to utilize that space, so somehow, now what happens in nanotechnology is that there is some effect of quantum mechanics that comes in, right? So uh, what is quantum mechanics? What is dual nature, right? that we can deal with somewhere else, maybe in future sessions. Let's just try and just try and understand it for a moment, that there is some level of physics that becomes very complicated, and that results in different properties that these materials start to show when they become nanomaterials. Right. So uh, if you go by the technical definition, a nanomaterial is one where at least one dimension should be less than 100 nanometers. All right. So if you look at the examples, and you can look at the comparison also, right? So here you have a football around 22 centimeter, and then you have flea 1 millimeter, right? And a red blood cell is around 7 micrometers, right? And then you reach the nanoscale level where you can see things like DNA, oligonucleotides, proteins, right? So, and then these are other nanostructures. All right. So uh, at nanoscale, you'll have different kinds of materials. Now these materials could be natural, like I showed you, DNA or proteins, and they could be artificial, like graphene. All right. This is the concept of nanotechnology, and if you want to just have a quick recap, what is the benefit? First is there's different properties, right? Right. So that is the one benefit. Then the reactivity is also more, right? Because if you the surface area will increase if you try and um, break a material into um, you know small particles. Then what happens is that actually what you're doing is you're increasing the reactivity, because then you're increasing the surface area that is available for reaction, right? So the reactivity will go up, and then there is quantum mechanics that is at play. Okay, right? So these are the three things, and then there is just the definition of nanomaterials, right? So then there's zero dimensional, one dimension, two dimension, three dimension, all these types of nanomaterials are there, right? So um, suppose it's one dimension, then it's mostly going to be a rod or a wire. If it's two dimension, it's going to be a sheet, right? So all these kinds of examples are there, right? So that's all about nanotechnology. Now, um, oh, let's try and try and link everything. Um, we have done space tech, and we've done um, biotech, and then we've looked at some kind of uh, nanomaterial nanomaterials, right? So there we were looking at the atomic and molecular levels. Like, so in space we were looking at large distances. In nanotechnology we are looking at smaller distances. So actually what happens is there are four fundamental forces that can govern the entire universe, right? So these forces are electromagnetism, gravity, strong nuclear force, and a weak nuclear force, right? So if you talk about the first two, they can operate at infinite distances. If you've done a little bit of physics, you know that formula, Gm when I put upon R square on for electromagnetic, it goes like Q1 Q2 upon R square, similar, something similar, right? So these forces can act at infinite distances, all right, and these act at smaller distances. Okay, okay. Now, strong nuclear force is usually the one that's you'll find inside the nucleus, where it's holding even the positively closed protons together, right? So that's a very strong nuclear force. Whereas when you look at the radioactive materials, mm, which are emitting particles, they are weak. They are exhibiting weak nuclear force, right? Right. So that's about the strong and weak nuclear force, and then you'll have electromagnetism, which will be a force between two charged particles, right? Okay. And then there's gravity, which is going to be effective between any two objects which have mass. Okay. Right. So, uh, electromagnetic force, gravitational force, strong nuclear force, and weak nuclear force are the four fundamental forces, right, that govern the universe. Now, what are the particles related to it, and you know, uh, which is the strongest? So, if we look at this, I don't want to go into the particles part of it because it becomes a little bit difficult over there, right? But if I just have to tell you what is the strongest is the strong nuclear force. No, because it's holding positively charged protons in a very small space. So that's the strongest force that's holding it over there. And if you have to look at the weakest, gravity is the weakest, right? Right. So those are the things about fundamental forces. Now, let's try and learn about a little bit of concept related to nuclear technology. Okay. So India has a three-stage nuclear program, right? And it's based on nuclear fission reactions. And we are working on different projects, which are based on nuclear fusion reactions as well, right? But for most of the purposes right now, when you're generating electricity, you're using nuclear fission. So what is nuclear fission? It is a nuclear reaction where the nucleus of a heavy element is split into lighter nuclei. So what happens is that initially you have a heavy nucleus like uranium, right? Then you bombard a neutron into this uranium, and then it will split into two lighter nuclei, which is krypton and barium, and it will give out three neutrons. These three neutrons can further be given to uranium, and again there will be a chain reaction. So there will be nine neutrons next, three here, and three here. So there'll be nine neutrons that will be produced, and then they can further chain the reaction. And this chain reaction, right, takes place over uranium-235 nucleus, right, and it keeps on happening, right? Now, because it keeps on happening, might have a valid question that okay, then it will be a very explosive reaction because at every stage you're also giving out energy, right? So it's going to be very explosive, and yes, it's true. But when you're trying to generate electricity using this, you usually try to control this reaction. And controlled nuclear fission reactions are already tested, tried and tested, and all the power plants are built on this principle. All the nuclear power plants, right? On the other hand, we have another type of nuclear reaction called a nuclear fusion reactions. Here, what happens is two lighter nuclei will come together and they'll form a heavier nuclei and they will give out energy and neutrons. Okay, okay, and uh, here, here again, this is a very very explosive reaction. And this reaction really cannot be controlled. Okay, so experimentally there have been demonstrations uh, of controlled nuclear fusion, but commercially we have not been able to produce. And imagine if we are able to produce this commercially, um, then we'll be able to produce a lot of energy at a very low cost. Um, and the reason for that will be that it needs, if you look at the things on the left hand side, these are isotopes of hydrogen. So those you can easily find in comparison to a limited resource like uranium, right? So uranium is in limited amounts, it's available in limited amounts. So that's not sustainable at all. But tomorrow, if you're able to control the nuclear fusion reactions, I mean, there's nothing better than that because there's ample amount of hydrogen available. Okay, so that's your nuclear fusion reactions. Actually, inside the sun also, if you remember when we were doing universal element formation over there, also I mentioned that nuclear fusion reactions are the ones which are taking place inside the sun. Remember? Okay. All right. So um, uh, a while ago, I spoke about electromagnetic forces, if you remember. Um, right. Electromagnetism. Now, what I like to do is like look at the electromagnetic spectrum. Okay, there's something called the electromagnetic spectrum, which is made up of electromagnetic waves. Now, these different types of waves have different characteristics. Okay, so you can learn this in an order, in any order, forwards, or if you want to even backwards. But this order is very important for a variety of applications, right? So you need to what are radio waves used for, what are microwaves used for, and those concepts will become easier if you're aware of this order, a particular order. So this particular order is arranged according to the wavelength or frequency. So if you look at the radio waves, they have the highest wavelength and the lowest frequency, while if you look at the gamma rays, they don't have as much of wavelength, but they're going to have high frequency and high frequency, and they'll also be having high energy. So you can look at the wavelength, it keeps on going down as you go to the right. If you look at the frequency, it keeps on going up, right? Now, where are we? We can only see this part of the spectrum with our eyes, which is the visible light, right? So that's the part that we can see, and other things which really cannot, right? Okay. Now here they are also mentioning the temperature of the objects at which the radiation is the most intense wavelength. Um, uh, the temperature of objects at which this radiation is the most intense wavelength emitted, right? So if you are basically increasing temperatures, right, that is when you are going to see these wavelengths. Okay. Now if you look at radio waves, microwaves, uh, radio waves are comparable to the size of buildings, microwaves are comparable to the size of humans or small insects. They're good for communication. So most of the times radio waves, that is why you call it a radio. Why? Because it's using radio waves. So radio waves, microwaves, they are mostly used for communication. Infrared is mostly used for heating purposes. Heating purposes. Visible light, you all know, is the light that helps us see around. Again, this can have applications in Wi-Fi. There's something called visible light communication. All right, so over there also, even visible light is being used for communication, right? Or there's a concept called Li-Fi, light fidelity, right? Right. So over there also, you can use visible light. In ultraviolet, you have telescopes, ultraviolet telescopes to look into outer space. Then again you have X-ray machines for examining bones, or you have X-ray telescopes for looking into outer space, and then same thing goes for gamma rays. Again, gamma rays could be used for treatment of cancers, right? For various kinds of therapies, research. So all these different waves, they have got different types of applications as well, right? So this is why knowing this entire spectrum is very important, because of the applications attached to this, right? So um, if you look at the relationship between wavelength and frequency, the higher the frequency, the shorter is the wavelength, and the lower the frequency, the longer is the wavelength. What is the wavelength? Wavelength is just the difference between these two tops or two bottoms, right? This is what is called as the wavelength. All right. And if I take if I take a column like this, a column like this, you'll find that there's only one wave over here, and there's at least three waves over here. So this is why the frequency here is higher. Okay, that is how you can understand. Okay, so that's the thing about the relationship between frequency, wavelength, and how when you go from gamma rays to radio waves, wavelength will increase, but the frequency will come. Um, uh, the frequency will come down, right? Yes. So the frequency at gamma rays is going to be the maximum. Okay, and so that will give them high energy also. They are very highly energetic waves. Okay, so that's mostly about electromagnetic spectrum. Um, Now let me. I was talking about communications. Uh, Let me just give you a brief idea of the generations of web. Okay. Um, So the communications and IT sector, it saw a boost with the coming of the internet, right? And the web, um, it had several generations. Okay, so we've already passed some of these generations, right? And it would be very easy for you to understand if you just go and look for old web pages. Those were just static web pages. You could not do anything with those except read them, right? So the goal essentially in the first generation of web was to share information. There was nothing else, right? You could just the simple web pages or you could write emails. The goal was just to share information. It was a static web, right? There was nothing that you could do, right? Just simple pages, no activity that you could do. Then came the dynamic web, which was the second generation. Here, the goal was not only to share content, but also to interact. So you could like, comment, share. So your Instagram, YouTube, Twitter, Facebook, all these allowed you to interact with the web by doing some kind of activity. Okay. And then right now what we are seeing is semantic web. Semantic web means that here we can talk to the web like we talk to a normal human. So here with the with things like ChatGPT or Deep Link, what you're trying to do is you're trying to make discovery of information more effective. You're trying to automate things, try to reuse things, basically making it easier, making it easier for the web to be understood by humans, right? So you can just type out a sentence and you can search, right? So that is the meaning of semantic web, okay, right? Okay. And the next stage that is predicted is called as the symbiotic web, where there'll be a symbiotic relationship between humans and machines. You'd find it difficult to separate these two. Now, but there's one more thing in Web 3.0, which is the concept of decentralized, private, and secure applications. So here, blockchain is an example of Web 3.0, which is a decentralized form of ledger, and it's considered to be a very secure form, very secure system of storing information, right? So, mm, as and when we move forward now in this Web 3.3 generation of web, you'll find that more and more things related to the semantic web will be visible. Okay, so those are the generations of web, okay, that we have. And lastly, let's look at um, the concept that's buzzing all around for the past 2 or 3 years. It's the use of this word has skyrocketed like anything, and that's AI. And you must be knowing already, you must be knowing the meaning of AI. It is a simulation of human intelligence by machines, especially computer systems. And they try and do something that humans are doing, that is called as artificial intelligence, right? And it allows machines to learn from experience, adapt to new inputs, and perform human-like tasks, right? So things like playing chess, self-driving cars, or chatbots are examples of AI, right? Because then what is machines doing? When they are learning from experience. When they're learning from experience, that's called as machine learning, right? So you show the machine 1,000 pictures of cats, and then you ask them to differentiate between a cat and a dog, and it will most likely be able to detect which picture is a cat. That is called as machine learning. You train them to recognize patterns, right, based on certain kinds of data and algorithm, right? Then you have something called neural networks, where you're trying to mimic or imitate the neurons in a brain. So connecting different sources, neurons in your brain, they are interlinked, right? When you're trying to do that, when computer systems are trying to do that, those are called as neural networks, right? Then you have something called natural language processing, and this means that the computers are able to process the natural language that we speak, right? So, like we speak in English, the ability to understand speech as well as understand and analyze documents uh, in a particular language, that is called as natural language processing, right? That's also an example of AI. And lastly, you have robotics, means that they are machines which can assist people with actual human, without actual human involvements, right? So those are your robots. So robots have been there for long. You can just program a robot to do certain tasks. Big machines in big factories. You'll find that if you are, suppose you have a packaging thing, so there'll be a robot which will just continuously be doing the packaging, right? So that's also an example of artificial intelligence. All right. So all these concepts that we have done, they pretty much form the essence of your entire foundation, right? And without this foundation, it's difficult to go ahead, right? So that is why I've tried to build on this foundation in this particular session. And as and when we move forward, we'll try and go a little bit deeper into concepts. And most of the concepts that we are going to cover will be from the news items, right? Either from the news items or from the important previous year questions, right? So that is going to be the approach for the future sessions. And yes, if you have any suggestions, you can always let us know. Subscribe to the channel for more such videos. And all the best, very best for your preparation. And yes, see you in the next session.